Okay, so welcome back to this next video in which we are discussing the SREBP pathway. Okay, so we're just in the process of discussing how cells can actually get a supply of cholesterol, and one of the key ways that they can get a supply of cholesterol is via the absorption of low-density lipoprotein particles. So we're just in the process of discussing the structure of a low-density lipoprotein particle. So we've discussed that the outer layer of a low-density lipoprotein particle is this phospholipid monolayer, and the main phospholipid that you use is a lecithin, also called phosphatidylcholine, which has a polar head which sticks out towards the watery environment, and then hydrophobic tails which stick in towards this hydrophobic core of the low-density lipoprotein particle. Now, within the phospholipid monolayer, you have a massive great protein, okay, and the reason that lipoproteins are called lipoproteins is because they have both a lipid component and also a protein component. So within the low-density lipoprotein structure, you have a massive great protein, basically, and this is the apolipoprotein B100. Okay, so there are many different apolipoproteins that are present within the different lipoproteins. Okay, but in low-density lipoprotein, you only have one apolipoprotein, which is a single apolipoprotein B100 protein. Okay, so these are massive great proteins. They dominate the phospholipid monolayer. Okay, right, and the reason it's called apolipoprotein B100, the apo thing is for, it's to denote that this protein is the protein that you find within the lipoprotein without the lipid component. So it means the protein without the lipid component, basically. Okay, right, and apolipoprotein B100 is usually just denoted apo B100, like so. Okay, so what are the other key components of low-density lipoproteins? Well, in low-density lipoproteins, you don't actually have a vast store of triacylglycerols. In other lipoproteins, such as very low-density lipoproteins and chylomicrons, in the lipid core here, the hydrophobic core of the lipoprotein, you would have a huge great store of triacylglycerols. However, low-density lipoproteins don't have many triacylglycerols in here, and the reason is that they are actually the remnants of the very low-density lipoproteins. So the very low-density lipoproteins circulate and deliver triacylglycerols to the skeletal and cardiac muscle during times of fasting, basically. And then once they've given away all of their triacylglycerols, they shrink hugely. They lose certain of their apolipoproteins, so that all that's left is an apolipoprotein B100. And in their core, they don't have any triacylglycerols, and they're then called a low-density lipoprotein. So low-density lipoproteins are the remnants of the very low-density lipoproteins. Okay, so what do you have within this core? Well, basically you have a huge number of cholesterol esters. So let's now discuss the structure of cholesterol, how we can create a cholesterol ester, and where cholesterol and cholesterol esters are going to be stored within the low-density lipoprotein. Okay, so let me firstly then show you the structure then of cholesterol. Okay, so cholesterol is a sterol, and sterols are based on steroid structure. So let me show you steroid structure. So steroids are actually defined chemically, which is something that shocks most people when they find it out for the first time. They expect steroids to be defined via their biological activity, but in fact, steroids are defined through their chemical structures. Okay, so all steroid molecules basically have a set of 4 carbon rings linked together. So they have 2 6-membered carbon rings, then a third 6-membered carbon ring, and then finally a 5-membered carbon ring here. So this is the basic core structure of a steroid. And whenever people draw steroid molecules, they always use skeletal formulae, because as you can imagine, if we were to draw this molecularly, it would look like a nightmare. Whereas when we draw it skeletally, when we don't show carbons, they're implicitly shown by corners, and we don't show hydrogens coming off carbons, it's extremely simple, because all a steroid molecule is carbon atoms joined to carbon atoms, and then hydrogen atoms coming off the carbon atoms, so it becomes extremely simple. Okay, now to turn a steroid structure like so into a sterol structure, what you do is you remove a hydrogen that comes off this carbon down here. Oh, and by the way, let's just go through a little bit of nomenclature so that I can point at carbons and actually have a number to say. So basically, the rings of the steroid structure are called ring A, ring B, ring C, and then ring D. Okay, and you can also number the carbons within the steroid structure. So you start with this carbon up here, so you call that carbon 1, you then go round carbon 2, carbon 3, carbon 4, carbon 5, carbon 6, carbon 7, carbon 8 is that one, carbon 9 is that one, carbon 10 is this one between A and B there. Then you continue on up here, so then we've got 11, 12, 13, 14, 15, 16, 17 up here. So this is the structure, well, this is a bit of nomenclature associated with steroid structures. And now that diagram's a bit cluttered, so I think to actually draw cholesterol I will grab a new diagram here. So let's redraw out our steroid structure, and now we can use some of our new nomenclature. So here is ring A, then we've got ring B attached onto ring A, which is another 6-membered ring. Then we've got ring C attached onto ring B, which is another 6-membered ring. And then finally ring D, which is the only 5-membered ring, attached onto ring C. Okay, right. So, to turn a steroid structure into a sterol structure, you take a hydrogen off carbon 3 of the steroid structure and you replace it with an alcohol group. So you're going to put an alcohol group on there instead, and that structure now is called a sterol structure. So any molecule which contains this sort of a structure is called a sterol. Any molecule which contains this sort of structure is called a steroid. So all sterols are steroids because they do have the steroid structure within them. Okay, right, so let's take cholesterol then. So, how do we convert the sterol structure now into cholesterol? Well, basically we stick a methyl group off this carbon here, carbon 10, with our numbering system. Okay, so we take the hydrogen that comes off carbon 10 off and put a methyl group there instead. Then we also go to carbon 13 up here, take the hydrogen that comes off carbon 13 off and replace it with a methyl group. Okay, then off carbon 17 up here we put a 7-membered side chain. So let me show this. So what we'll have is 1, 2, 3, 4, 5, 6, 7. A 7-membered side chain, and it's attached to the second carbon of the 7-membered side chain. Okay, and then you also have a methyl group off the 6th carbon of the 7-membered side chain. So, the numbering system now for, one more little modification to make for cholesterol. You also have to turn that into a double bond there. So take the hydrogen off this carbon, take the hydrogen off this carbon, create a double bond between them. Okay, right. So, updating our numbering system now, because we continue with this numbering system, so all the carbons of the steroid structure are still named the same, but we now have a few extra carbons to name. Okay, so we start with this one. So this methyl carbon here is called carbon 18, so we're continuing on. So this is 17, this is 18, this one then is 19, this one is then 20, then this one 21 here, and then you go on. So this is 22, 23, 24, 25, 26, 27 basically. I'm not going to try and squeeze that in there because there's not really enough space. So this is 27, this is 26, 25, 24, 23, 22. Okay, right, so that's the structure then of cholesterol. So this is free cholesterol. Now you do actually have quite a lot of free cholesterol molecules within the low density lipoprotein. However, these cholesterol molecules, the steroid structure here, this is extremely hydrophobic. So this doesn't interact well with water at all. However, you do have one little glimpse of polarity over here in the form of this alcohol group that's over here. So basically, these are like phospholipids. They can stick they're not phospholipids, obviously. They don't have a phosphate group or a long-chain carboxylic acid within them, but they're like the phospholipids in that they have a polar portion and then a hydrophobic portion. So actually, these cholesterol molecules can sit within the phospholipid monolayer. So free cholesterol will be present from here to there in the phospholipid monolayer, basically, and they'll be oriented such that the alcohol group that is free faces out into the outside world, basically, where there is lots of water, whilst the neutral hydrophobic ring structure, that faces into the middle, basically. So then let's just complete up our phospholipid monolayer by having lots of phospholipids everywhere else. So basically, this phospholipid monolayer consists of mainly phosphatidylcholine phospholipids, then with a few free cholesterol molecules dabbed in from place to place. And then you have this apolipoprotein B100 dominating the outer surface. Now, in the core of the lipoprotein, what you mainly have is cholesterol esters. So let me explain what a cholesterol ester is. So let's now turn our attention from cholesterol to what a cholesterol ester is. Okay, so basically, to turn a cholesterol molecule into a cholesterol ester, what you're going to do is you're going to bring a long-chain carboxylic acid in over here. So here is our long-chain carboxylic acid, and what we're going to do is we're going to esterify this long-chain carboxylic acid onto that alcohol group of the cholesterol molecule, like so. So you'll take the hydrogen off the alcohol group of the cholesterol molecule, you'll take the alcohol group of the long-chain carboxylic acid, you'll then link this carbon of the long-chain carboxylic acid to the oxygen of the alcohol group to create a ester link here, and this is now called a cholesterol ester. Now, what does this mean? Well, it means that the cholesterol molecule had this one glimpse of polarity over here in the form of the alcohol group. That's now gone. Okay, so in cholesterol esters, you don't even have that polar group over here. Instead, you have a great big hydrophobic group sticking on over here. So these molecules are truly hydrophobic, not even a glimpse of polarity anywhere anymore. Okay, so these molecules cannot be put in the phospholipid monolayer because they don't have a single group that can be oriented so that it's sticking out to face the water. So these molecules need to be in the hydrophobic core of the low-density lipoprotein. So basically, let's draw these in now in the hydrophobic core. So I'll just draw the cholesterol molecule as a blue box, and then I'll draw the long-chain carboxylic acid that is sticking off the cholesterol molecule to create the cholesterol ester. I'll draw that as an orange line here. So there is our cholesterol ester sitting in the hydrophobic core, and of course you won't just have one, you'll have many of them in there. Okay, so that's now the structure of a low-density lipoprotein molecule. Now, because the main cargo of low-density lipoproteins is the cholesterol molecules and the cholesterol esters, you will often hear people calling low-density lipoprotein molecules merely as cholesterol, okay, because that's their main cargo, that's what they're transporting around. All lipoproteins have a apolipoprotein and a phospholipid monolayer containing a lot of lecithin. But what makes these special is that their main cargo then in the hydrophobic core and also in the phospholipid monolayer is free cholesterol molecules and cholesterol esters. Okay, so that's why people often refer to these molecules as cholesterol. Okay, so there's an introduction to low-density lipoprotein. What we now want to see is how do cells take up low-density lipoprotein. Okay, and firstly, let's just give the motivation for why they would want to. Well, basically, cholesterol has been given a very bad reputation by the media. The media portray it as this terrible, terrible molecule, when in fact it's actually an essential part of the cell membrane. So all cells need a certain amount of cholesterol within their cell membranes. Okay, so cells will take it up so that they've got this cholesterol for their cell membranes. In addition, the cells of the adrenal cortex use cholesterol to synthesize steroid hormones such as aldosterone, cortisol is another example, the sex hormones such as testosterone, estrogens, progestogens, all of those are based on cholesterol molecules initially. Okay. In addition, the liver uses cholesterol as a major component of bile. It uses cholesterol to produce the bile acids, which are important in forming mixed micelles and helping in the absorption of lipid molecules within the small intestine. Okay, so cholesterol is not all bad, so there are good reasons why a cell would want to uptake cholesterol, basically. So let's now talk about the way that they can uptake it from the blood in the form of these low-density lipoproteins. Okay, so basically this requires the Ldl receptor. Okay, so what I want to now draw is the trafficking process that the Ldl receptor goes through. So let's let this be the nucleus here. Okay. And then basically we have this large intracellular organelle which is responsible for much of the protein production, basically. Okay, especially proteins that are going to be put on the plasma membrane. So basically, the Ldl receptor starts its life within the rough endoplasmic reticulum, which is this intracellular organelle that I've drawn here. So this balloon thing here represents the rough endoplasmic reticulum, and for short, the rough endoplasmic reticulum is usually abbreviated to little r and then capital E, capital R. So the little r is for rough, then the capital E is for endoplasmic, and then the capital R is for reticulum. Okay, right, so here is our Ldl receptor here, and that little blue bit that I've put there is supposed to represent the ligand binding domain, which will actually bind the LDL molecule. Okay, so this represents the LDL for low density lipoprotein, and then I'll put an R for receptor. Right, so it starts its life in the rough endoplasmic reticulum, and hopefully what you've noticed here is that the ligand binding domain is facing into the lumen of the rough endoplasmic reticulum, whilst the portion that will eventually be the cytoplasmic domain at the plasma membrane, that's nicely facing into the cytoplasm, so that's all good. Okay, basically the lumen, the stuff that's on the luminal side of the ER membrane, that is the portion that will eventually face the outside world. Okay, right. So once you have synthesized the LDL receptor within the rough endoplasmic reticulum, what happens is that this will be shipped from the rough endoplasmic reticulum to the Golgi apparatus. So let me draw the Golgi apparatus here. So the Golgi apparatus is also a membrane-bound organelle, and it consists of around 7 structures called cisternae. Okay, and what these cisternae are is basically they are membrane-bound compartments which are in the shape of discs, basically. So imagine taking a sphere. Okay, so imagine taking like a sphere, and then, so it's like a balloon surface, and then sort of squashing it down, so that what you end up with is like a disc. Okay, that's what these basically are, and then you stack multiple of these discs on top of one another. Okay, and generally you have around 7 of these things stacked on top of one another in a Golgi apparatus. So here we go, here is our Golgi, and they gradually get smaller as you go along. So this is the Golgi apparatus. Okay, now, when you deliver contents from the rough endoplasmic reticulum to the Golgi apparatus, you come to this side which faces the rough endoplasmic reticulum first. Okay, and the side which faces the rough endoplasmic reticulum is called the Sis Golgi. So cis means on the same side as. Okay, so this is the side of the Golgi which is on the same side as the rough endoplasmic reticulum. Whereas this other side of the Golgi, which faces the plasma membrane, well, which is closer to the plasma membrane, this is known as the trans Golgi. Okay, and this trans means on the opposite side. Okay, so this is on the opposite side of the Golgi to the side which faces the rough endoplasmic reticulum. Okay, so what's going to happen is that little vesicles are going to bud off the rough endoplasmic reticulum like so. So here we have a little vesicle, and these vesicles will contain the LDL receptor. Okay, so here is the LDL receptor with its ligand binding domain there in blue. Okay, and these vesicles here that move between the rough endoplasmic reticulum and the Golgi apparatus, specifically the Sis Golgi, these are called COP2 coated vesicles, and we will discuss COP2 coated vesicles in much more detail later when we actually got onto the SREBP pathway. Okay, right. So COP2 coated vesicles. Okay, right. So we'll discuss that in more detail later, but for now it's fine to just imagine a vesicle pinching off there and taking the Ldl receptor with it, and it's then going to take this to the Sis Golgi. It will fuse with the Sis Golgi membrane, and what you'll end up with then is the Ldl receptor facing into the lumen of the Golgi. So the ligand binding domain here in blue is facing into the lumen of the Golgi. What will then gradually happen is this Ldl receptor will be transported from cisterna to cisterna to cisterna from the Sis Golgi to the trans Golgi over here, which I'll try drawing like so. Okay, so here is our Ldl receptor. Right, and as it goes through this process, moving from the cisterna to cisterna to cisterna, it will gradually get a bunch of changes happening to it. So it will have a huge bunch of modifications occurring to it, post-translational modifications. Okay, then what will happen is a vesicle will bud off from this trans Golgi like so. So here we have a vesicle which will have our Ldl receptor within it. Okay, and here's the ligand binding domain still facing into the lumen of the vesicle, so that the cytoplasmic tail is then already in the cytoplasm. Okay, and this sort of a vesicle that buds off from the trans Golgi is called a secretory vesicle. Okay, now what will happen is this secretory vesicle will take this Ldl receptor to the plasma membrane. Okay, so it's now going to go to the plasma membrane. So let's draw the plasma membrane here, and here comes our secretory vesicle with the Ldl receptor inside it here. And I'll draw that ligand binding domain of the Ldl receptor in blue here. Right, okay, what will then happen is this secretory vesicle will fuse with the plasma membrane, and what will happen then, since I've got more space now, I can actually show the fusion. So here is the vesicle fusing with the plasma membrane, okay, and you can see that what's going to end up happening is that this Ldl receptor will end up with its ligand binding domain on the extracellular side. Okay, so just to highlight this further, this is the extracellular fluid side, the ECF, and this is the cytoplasmic side on this side. Okay, and the overall result of this is that you've then got the plasma membrane here, then with the Ldl receptor on its surface like so, and the ligand binding domain is facing the extracellular fluid. Okay, so that's how we get the Ldl receptor to the plasma membrane. Let's now discuss what's going to happen when the LDL actually binds to the Ldl receptor. So, here is our plasma membrane with the Ldl receptor on its surface. Okay, like so. And here's its ligand binding domain. So I've now turn this picture around 90 degrees. So this is the extracellular fluid, and then this is the cytoplasmic side here. Okay, right. And what's going to happen is our LDL molecule is going to come along and bind to the extracellular fluid side of this Ldl receptor. Okay, so specifically it's the apolipoprotein B100 of the LDL molecule which binds to the ligand binding domain of the Ldl receptor. Okay, and there's certain interactions which are very important in the binding of apolipoprotein B100 to the ligand binding domain of the Ldl receptor. Specifically, in the ligand binding domain of the Ldl receptor, what you have is a great number of acidic residues. Okay, so let me just highlight this. So within this ligand binding domain of the Ldl receptor, you have acidic residues. Okay, and these are going to bind to basic residues which you have present in the apolipoprotein B100. So how does this work? Well, basically, the acidic residues, those are acidic because they can donate protons away into the solution, basically. Okay, so these acidic residues will donate protons away into the solution. And let me give you an example of an acidic residue because it will help. Okay, so if I draw the example, and I'll take as an example aspartic acid. So I'll draw it as though it's a residue within a protein. So here's the amino group. Here's the alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group, which will be linked to the amino group of the next amino acid along. And then the R group of an aspartic acid amino acid is a methylene group, then with a carboxylic acid group on the side. So this is aspartic acid. Okay, so it's an example of an acidic amino acid residue. So what aspartic acid can do is it can donate this proton off from that alcohol group. So the proton can fall off, leaving a negative charge on that oxygen atom of the alcohol group of the carboxylic acid group. Okay, and therefore it is an acid because it has donated a proton away into the solution. Now, if we have a huge number of acidic residues within the Ldl receptor ligand binding domain, then some of them at any one time will donate away their protons and will now be in their conjugate base form and have a negative charge. Okay, so basically the message is that this Ldl receptor ligand binding domain will have negative charge, basically. Meanwhile, if we look at an example of a basic residue, and the archetypal example of a basic residue will be a lysine residue. Okay, so again, here is the core amino acid structure with the amino group, the alpha carbon, the carboxylic acid group, and then the R group consists of 4 methylene groups. So here's a methylene group, and we'll have 4 of those with an amino group right on the end. Okay, now this nitrogen of the amino group here has a lone pair of electrons, okay, and this lone pair of electrons serves as a fantastic docking point for protons within the solution. So protons within solution can come and bind to this lone pair of electrons, which will give that amino group an extra proton and therefore a positive charge. So basic residues basically can gain protons and therefore become positively charged. So the overall message is that residues within the Ldl receptor ligand binding domain are going to become negatively charged, whilst residues within the apolipoprotein B100, which are basic, are going to become positively charged, and the positive and negative charges combine to one another, and that's how the LDL molecule binds to the Ldl receptor. Okay, right, so what now happens once the LDL molecule has bound to the LDL receptor? Well, basically, within the cytoplasmic tail of the LDL receptor, you have a special motif known as the NPXY motif. Okay, so this is the NPXY domain, and then we'll have the carboxylic acid terminus of the protein down here. Now, what is this NPXY motif here? Well, basically, this is what's known as an endocytic motif. Okay, so it promotes the endocytosis of this LDL receptor. Right, and basically it's a sequence of 4 amino acids that is within this carboxylic acid tail, which can be recognized by proteins within the cytoplasm of the cell as meaning endocytose me, basically. This is the signal to proteins within the cell to endocytose this receptor. So basically, when LDL binds to the LDL receptor, some sort of conformational change will occur that allows this signal here, this NPXY motif, to become visible, and it will then be able to interact with proteins which are in the cytoplasm, which will then begin the endocytosis. Okay, so let's just discuss what these 4 amino acids are, okay, that make this endocytic NPXY motif. So to understand this endocytic motif, you need to be able to read the single letter amino acid code. So basically, N means asparagine. So the first amino acid within this endocytic motif is going to be an asparagine residue. So let me show you the structure of an asparagine residue. So again, I'll draw it as though it's a residue, so I'll draw it as though it's within a protein. So here's the amino group, here is the alpha carbon, here is the carboxylic acid group, and the R group of asparagine basically consists of aspartic acid, which we've just seen up here, with a primary amide group. So take this alcohol group off and put an amino group on there instead. That's asparagine. Okay, so here's the methylene group, and now here is our primary amide group. So here is the amino group coming off that carboxylic acid group. So this is asparagine. Okay, then the second amino acid within this NPXY motif is going to be a proline. So P is for proline. Now proline is the unusual amino acid because its R group involves the amino group. So in proline you're not going to have a hydrogen coming off this nitrogen. Instead you're going to have that nitrogen involved in the R group. So off the alpha carbon you still have a hydrogen, however, and basically now what we're going to have is a 5-membered ring formed here. Okay, and rather than having the carbon and actually drawing the 2 hydrogens off, I've just put CH2, but what that means is you have a carbon here with 2 hydrogens coming off. So basically you've got 3 carbons here that form this 5-membered ring here, and each of these carbons has 2 hydrogens coming off, so that's a proline residue there. Okay, then the third amino acid along basically can be any old amino acid. This is X now, so X means it can be any old amino acid. It's not a set amino acid. Okay, and basically to denote that I'll just have the R group as an R to denote that it's arbitrary there, which amino acid you put there. Okay, and then the final amino acid you have is Y, which stands for tyrosine. Okay, so this is tyrosine. So the single letter amino acid code Y means tyrosine. So here's the amino group, here's the alpha carbon with a hydrogen coming off it, here's the carboxylic acid group, and then it will be linked to the amino group of the next amino acid along. Now the R group of tyrosine basically consists of a methylene group, then with a benzene ring coming off, which I'll just draw like so. So here is the benzene ring, and then we've got an alcohol group coming off that benzene ring here. So that then is tyrosine. So basically in this endocytic motif, NPXY, what you have is asparagine followed by proline followed by any old amino acid, so this just means X, which can be anything. Okay, so any of the 20 amino acids. Okay, and then we also have tyrosine Y. Okay, so if you have one of these motifs, it doesn't matter what this middle amino acid is. If you have an asparagine followed by proline followed by something followed by a tyrosine, that will tell proteins within the cytoplasm to endocytose you. So something about the binding of LDL to the Ldl receptor will change the exposure of this NPXY motif so that it can now interact with downstream proteins to cause the endocytosis of the Ldl receptor. Okay, so what's now going to happen is another protein is going to come and bind to this NPXY endocytic motif, and this next protein is an adaptor protein. Okay, and it's called an adaptor protein because it's basically just an adapter. It will bind to the Ldl receptor, and then it will also bind to the other protein which is going to trigger the endocytosis. So really it just is a linker between the receptor on one hand and then the endocytic machinery on the other hand. Okay, right, so what is this adaptor protein called? Well, basically it's called the LDL, that makes sense, receptor, that also makes sense, so the LDL receptor, and then it's the adaptor protein 1, so AP stands for adaptor protein 1, so this is short for low density lipoprotein, that's the LDL, and then receptor, and then adaptor protein 1. Now, there is another name for the low-density lipoprotein receptor adaptor protein 1. It's also called the autosomal recessive hypercholesterolemia protein. Okay, and that's because it's found to be involved in autosomal recessive hypercholesterolemia. Specifically, if you have double hit mutation on your 2 genes for this low density lipoprotein receptor adaptor protein 1, then you can't remove LDL molecules from the blood, so LDL remains in the blood and you get massively hypercholesterolemic blood. I think it can go up to like 204 millimolar or something awful like that. It's supposed to be around 3, okay, so that it's not a nice disease to have. So, this protein is also known as the autosomal recessive hypercholesterolemia protein. Okay, and for short, the autosomal recessive hypercholesterolemia. Oh dear, I'm not going to be able to fit hypercholesterolemia in here. A massive great word. For short, the autosomal recessive hypercholesterolemia protein is usually abbreviated to ARH. Okay, so this is ARH. So you might see people call the Ldl Receptor Adaptor Protein 1 the ARH protein, and that's just standing for Autosomal Recessive Hypercholesterolemia protein. Now, what does this protein do? Well, basically, it's going to come and bind to this MPXY motif. So it's going to come and bind here, basically. So I might denote it as the autosomal recessive hypercholesterolemia protein, just because that's shorter. Okay, and then what the autosomal recessive hypercholesterolemia protein is also then going to bind to is clathrin. Okay, and clathrin is then going to trigger the endocytosis of this Ldl receptor along with its cargo. Okay, and we'll continue this discussion where we'll talk about clathrin-mediated endocytosis in the next video.